Welcome back. This is the next part of our conversation with John on how to digitize biobank operations using barcode technology. And we are picking up exactly where we left off. So let's jump straight back in. John, once you are listed the requirements for the labels, software, printers, scanners, um, let's move to selecting the label providers. What are the considerations when selecting a label provider?
After you've outlined your specific requirements, the next step is to research different labeling providers that can meet those needs. When evaluating these providers, you want to consider several factors to ensure that they're the right fit for your biobank.
Yeah, I agree, John. There are several important factors to consider when purchasing labels from a label provider. Do they have experience working with biobanks or other biomedical laboratories to understand the processes that and the requirements of such facilities? And the other factor would be their capabilities on customization. Very often, the biobank or biomedical lab or biotech company would need some customization of labels, which requires the provider to have the right equipment, the right tooling to be able to do this type of things, and also to understand the materials that they are using for making labels for such applications. Uh, Recently, I wrote an article on how to select a label provider for biobanks. I'll put the link in the description.
I'm sure that would be very helpful.
John, once you shortlisted the label providers, do you recommend that testing of the labels conducted in the lab by a biobank?
So, once you've shortlisted your labeling providers that you think best meets your needs, the next step is to request product samples from them and test the system on a small scale to ensure that it meets your biobank's requirements. This pilot testing is a very important step before you fully implement the barcode solution across your entire lab.
And how would you do the pilot testing, John?
You need to reach out to your shortlisted vendors, explain your specific requirements, and seek their assistance in selecting the best products from their offerings that best meets your needs. Then comes the most important step. Request samples of the recommended products for testing. A reputable provider should have no issue providing free samples for evaluation. They should also provide detailed data sheets for their labels. Never buy labels only on the website description or catalog details. Always request samples first and test them in your actual lab environment. And once you've received your samples, select a representative subset of your workflow for pilot testing. Label a small batch of samples with the provided label samples and monitor the label's adhesion, durability, and readability under your specific storage conditions. If you encounter any issues, such as the labels peeling off, becoming illegible, or failing to scan correctly, contact the solution provider and make the necessary adjustments, such as selecting different labels or adjusting the application technique to ensure optimal performance. Make sure that the labels meet all the requirements and perform as intended in your particular lab environment.
I agree, John. Pilot testing is highly important, and and once you conducted the pilot testing in a lab and you approve the label, and subsequently there is a change, for example, in a container brand, or now you are sourcing it from another supplier, it's always a good idea to redo the testing with the new containers because although they might look the same or based on data sheet they might be made from similar materials, it might impact how the labels perform on that particular container. I have seen many cases, for example, with cryo vials, that a lab was purchasing one cryo vial, and subsequently they decided to purchase another one. And both are made from polypropylene, and supposedly should perform the same way, but actually we found that the same label would perform very well in cryo temperatures on one vial and wouldn't perform as well on the other one. So it's a very good idea to try to retest when there's a change in a container that you use in a biobank. It's also important to mention that selecting a ribbon that is compatible with labels is also very important. There are large variety of ribbons available. They fall mainly in three categories, like resin, wax resin, and wax, and resin being the most durable one. But there's a huge variety of ink formulation in each category. Therefore, selecting the right ink formulation or the right ribbon for your end use application is as important as the label itself. John, now what comes next after the pilot testing is completed?
Once pilot testing is successful and any potential issues have been addressed, you can proceed with a full-scale implementation of the barcode solution across your biobank.
John, what are the steps for full-scale implementation of barcode technology in a biobank?
The approach to full-scale implementation can vary depending on your lab's quality assurance requirements. For some, it might be as straightforward as printing the labels and applying them to samples. For others, it may involve a more comprehensive strategy to ensure everything is up to standard. Let me walk our viewers through a few key steps for a smooth and successful implementation. First, I strongly recommend that you have an implementation plan with set timeline, budget, resource allocation, team members involved, and their roles and responsibilities for the project. Next, it's preferable to assign dedicated personnel to oversee different aspects of implementation. For example, designate staff responsible for applying the labels onto the samples, for training the lab staff, and those who will handle the technical integration with LIMS or the data management platform, if any. Then make sure you have the necessary labels, printers, and scanners in place before starting the implementation. If needed, integrate barcode software and label printer with your LIMS to directly print from within the LIMS and ensure automated sample tracking. Next, redesign your sample management workflow with barcode scanning integrated at key points in your sample processing pipeline, ensuring efficient data capture and minimizing disruptions to existing processes. You may want to implement scanning at these key points: the point of sample collection, the start of processing, just before storage, storage location changes, and sample retrieval, etc. Conduct training sessions for all staff members who will be using the barcode system. Ensure they understand how to print and apply labels, how to use barcode scanners, and how to log information into the LIMS. Training should also cover troubleshooting common issues and maintaining the equipment.
Training the staff is highly important to avoid any unpleasant surprises. I remember a case that I received a phone call from a lab, and they were complaining that they had these labels that they purchased somewhere. And then when they put the labeled vials into autoclave, when they opened the autoclave, all the information was gone and the labels were completely black. There's no information, identification of the vials. And when I start asking what type of printer they have or type of labels, it turned out they were using direct thermal labels. These labels are not designed to go into autoclave. They are actually, the principle behind their printing is that when they are heated, they turn black, and that's how they print. So putting them in autoclave obviously was expected to turn them black. Apparently someone was not trained who was doing the labeling of their vials. In another instance, again, I received a call from a lab that they were doing histology, and the slides that they had, the patient information, the information was gone when they put it through a histostaining process. And apparently they didn't use specialized ribbons or labels that are designed to withstand harsh solvents like xylene and alcohol gradients and so on. And the information was lost. Obviously, these are very unpleasant type of situations that can be avoided by simply training your staff properly on labeling, and your label provider should be the one of the sources where you can get that information or even the training for your stuff.
That's why the staff should not only have the adequate practical knowledge about label application, but also the technical know-how of labeling technologies available to them. Once everything is in place, proceed with applying the barcode uh, system across your labels systematically. Make sure your samples are properly labeled according to your predefined standards. Gradually update your lab's standard operating procedures with a new workflow. Standard operating procedures are the backbone of any successful implementation. You'll need to create or update your SOPs to document the new barcode-enabled workflow. These SOPs should provide clear instructions for sample handling, label application, barcode scanning, personnel responsibilities, and procedures for handling errors or exceptions.
That's true, John. The SOPs are not static documents. They need to be regularly reviewed and updated to meet the evolving needs of your lab. Moving on, John, is there a need to monitor the barcode system, um, how it works for a biobank?
Absolutely, George. After full-scale implementation, ongoing monitoring and evaluation are essential to ensure the barcode system continues to meet your biobank's needs. Conduct regular audits of the workflow to ensure labels are adhering correctly, barcodes are scanning accurately, and the data is being logged properly. Encourage your staff to report any issues or suggest improvements. This feedback is crucial for identifying areas that need adjustment.
That is absolutely true, John. The team's feedback is very important. If they notice any discrepancies or any issues that come up with, for example, with adhesion of labels or printing on the labels, they should immediately bring up to their provider, a label provider, ribbon provider, so they can be addressed in a timely fashion. So the any issues that are there it doesn't continue and cause further issues down the road. For example, that after maybe a few weeks or a month of storage to find out that the labels fell off, for example. Okay, John, can we now a little bit talk about the challenges that biobanks may have during implementation of the barcode technology?
Absolutely. Implementing barcode technology in a biobank can certainly present a few challenges. The number one challenge is that biobanks who are implementing barcode technology face is that the staff needs time to adjust to adopting to the new technology due to the comfort with their existing processes. For years, they might have been labeling samples manually and logging the information in physical notebooks or Excel sheets. So transitioning to a digital system can seem daunting at first.
And what's the solution there, John?
To overcome this, involve staff early in the planning process, provide comprehensive training, and highlight the benefits of the new system, such as improved accuracy and efficiency, to get their buy in.
It's true, John. My experience in this, if the team is involved into the process of implementing the barcode technology and they can see for themselves how it's going to make their life easier down the road, I think they would have no problem to be on board with implementing the new technology in their laboratory.
Another challenge could be that integrating barcode technology with your existing LIMS or other sample management systems can sometimes be a bit complex. Ideally, you want to seamlessly record sample data into your LIMS and print the labels directly from it. When you create labels, you often use templates that define what information is included, how it's arranged, and the overall design of the label. If your LIMS doesn't support basic label templates that you plan to use, for example, if they can't handle the layout or data fields required for your barcodes, it could lead to difficulties in printing the labels correctly. Similarly, if the LIMS isn't compatible with your planned printing setup, perhaps it doesn't integrate well with the printer you're using or doesn't support automated printing processes, this could create issues in efficiently producing the labels you need. The solution is to work closely with your LIMS solution provider to ensure seamless integration. Conduct thorough testing to identify and resolve any compatibility issues before full-scale implementation.
I believe, John, also selecting the right label for your biobank is also a challenge, isn't it?
Agreed. With so many label options such as direct thermal, thermal transfer, laserjet, inkjet, permanent and removable labels, and from so many different vendors, it could be challenging to determine which labels best suit your specific needs. But if you keep in mind the points we discussed today and work closely with your labeling solution provider, you will not face any problems in selecting the right labels. Just as we work closely with our clients, for instance, I could give you an example of a biobank partner who was looking for a solution to implement in their new biobank collection. So they've accumulated a whole set of biobank samples, and they were looking to have an efficient system installed. But what I noticed that they didn't want to thaw out their samples before labeling, before applying the labels. So I noticed that they wanted to preserve the frozen nature of the samples. So I quickly discovered that they could use a label that is specifically designed to be applied onto an already frozen vial. And that really is ideal because they do not have to jeopardize the biological integrity of their sample by thawing it first. And I must say, they were really impressed with that solution.
John, that's a good point. On many occasions, I received also requests for such a label, and there is such a label that I think it's called cryostock that can be applied at frozen temperatures and then stored inside liquid nitrogen. And many biobanks use this type of method to re-identify their samples without the need to thaw them. John, one key takeaway from the session, what would be that?
If there's one key takeaway for our listeners today, it's this. Make sure to work closely with your label provider and conduct thorough pilot testing before moving on to full-scale implementation. It helps identify any potential issues and allows for adjustments to be made early on, ensuring a smoother transition and more reliable results. Partner with a provider who has experience working with other biobanks and offers testing facilities to validate the labels in the actual lab setting.
Thank you, John. This has been very informative session. I'm sure the information you shared with us would be very helpful for biobank leaders who are looking forward to or planning to implement Barcode technology in their facilities. And to our viewers, thank you for tuning in to Lab Leaders Edge. If you have any questions or topics you'd like us to cover in future episodes, feel free to reach out to me or John on LinkedIn or visit labtalk.com. See you next time.